very slow. Okay. What I'm going to talk about is a few open questions in human evolution, which could benefit from molecular insight and some of the contributions that paleoproteomics has already given to that. Then we'll shift a bit to problems and hurdles that we are expected to face and the practical and ethical considerations around them, focusing on the potential power of paleoproteomics, so asking what is answerable with paleoproteomics and what is not, and finally kind of suggest a possible path forward. So you might be asking with such big question, who is speaking? Who is it? So hi, I'm Oded. Most of you have not met me yet. I recently joined as a postdoc to Pontus Skoglund's lab at the Francis Crick here. Before this, I was doing my PhD in chemistry. And I was developing drugs that were part protein, part synthetic molecule, to target Alzheimer's and Parkinson's disease. And you might be asking, what is the relationship between these two things? What is the connection? So I kind of want to draw where I feel more confident and where you maybe I'm not qualified to speak about. So some things I am qualified to speak about maybe are post-translational modifications of proteins, both causing them intentionally and detecting them using mass spectrometry. That also taps into previous knowledge from my master's, where I was working in a mass spec lab on redox chaperones. So a lot of mass spec and also focusing on protein degradation and how proteins evolve over time. And in my ancient past, I was doing mathematics. I was a software engineer in the tech industry. And that will connect to some of the things that I will say about database design and about statistics and probability. What I'm not, at least not yet, is any kind of archaeologist, bioarchaeologist, or any kind of anthropologist. So take with a big grain of salt anything I say on these topics. I'm not a geneticist or a population genomics expert, as will be evident. I'm also not a bioethicist or philosopher, despite what my slides may suggest. And with that, I want to jump straight into human evolution. I don't think this crowd needs the introduction of the hominin tree, but what I do want to focus on is where do we have molecular evidence, and in this case, DNA evidence. For most of it, we have no DNA at all. Hundreds of thousands to a bit over a million years, we could theoretically get DNA evidence, but humans were not in the areas where we can. So there is a big gap of molecular evidence in human evolution. And there are open questions which could really benefit from molecular evidence, like the interactions between the different hominin lineages and the origin, place, and time of origin of our own species. And this is where paleoproteomics can come in. So as you all know, proteins also encode genetic information and survive much longer. They carry out information from the DNA all the way down to protein, but we can find them even in the subtropics, hundreds of thousands, even millions of years. And then we can kind of reverse engineer the information that we find in proteins to what DNA changes caused them. The problem in this is that there is major information loss in this process. So if we look from DNA to RNA, only 1% of the genome is protein coding. And if we look at a specific tissue, we only get a tiny fraction of the proteins that are actually there. Then what we see in proteins are only non-synonymous mutations. So even if we have SNPs in the coding sequence of a protein, it doesn't necessarily mean we will see them in a protein, in the final protein product. And that is true for any proteomic experiment. Then when we look at ancient samples, we also have degradation, evolutionary conservation of collagens, contamination, all the things that were mentioned today in the second session. Despite this community has published incredible things doing phylogeny with paleoproteomics, from fauna and getting closer and closer with Gigantopithecus and Paranthropus to Denisovans. So we are making strides towards studying human evolution with paleoproteomics. I do want to kind of point out some of the hurdles we might be facing going forward, or we can expect to face. One of them is the use of diagnostic single amino acid polymorphism, SAPs. If we look at a mutation that is unique to the denisovan, so we only find it in genomic sequences of the one denisovan we have, and we find a sample that has that unique polymorphism, it is usually used to assign it to be the denisovan, but there is some probability that it is not, or that it has part denisovan ancestry and part Homo sapiens ancestry. This is even more true if we look at a derived mutation in the human reference proteome. So if we look at the human reference proteome and find a mutation that is unique there compared to the ancestral variant in our close relatives, it is very important to look at the variation within Homo sapiens, because when we do that, we often find that there is variation. Sometimes it's small, sometimes it's big. Sometimes the reference proteome actually represents a minority position, especially in the collagens. So this is very important to not fall into these pitfalls. I think we've kind of mentioned this throughout the day, the responsible use of the fossil record and how what are the ethics behind using and destroying samples for paleoproteomics. And I think that this discussion is really important. We need to discuss it. We also need to have as much information as we can to discuss it intellectually and to have a real informed discussion on the topic, because the samples are the most precious resource. We can spend a lot of time preparing and discussing the background of it. And I think the most important question in order to discuss ethics is what is possible? What can we do with paleoproteomics? So with ancient DNA, we can give individual level ancestry for humans. We've seen today talks about zooms, so we have family and genus level identification of species. Where is the full potential of paleoproteomics? It's somewhere in between those two, but the question of where is really important. And in order to inform that, and kind of drawing inspiration from Palaeoprofiler that was published last year, I am building a dynamic database that links genomic and proteomic data dynamically. So it's not like a static translation of genomic data into proteomic data, but rather we take SNPs based on modern human big genomic projects like the 1000 Genome Project, as well as ancient genomes. That informs the genomic side of the database, and taking ancient proteomic samples to inform the proteomic side of the database, and then connecting them dynamically so we can predict SAPs from SNPs, and we can look at SAPs that we do detect in paleoproteomics and kind of extrapolate which SNPs may have caused them or are likely to have caused them. Using the data generated by this, we can kind of have, this is a very preliminary estimation of what is possible with paleoproteomics in the context of human evolution. So here I'm looking at the data that was derived from the two Tibetan denisovans, suspected denisovans. And the actual observed, if we look at the actual observed sequence and the number of non-singleton SAPs in the human proteome that it covers only a couple dozen. This is not enough to tell apart Neanderthals and modern humans, and barely enough to tell apart denisovans and modern humans. If on the other extreme, we look at all the proteins that have ever been detected in bone and look at all the non-singleton SAPs in them, there are tens of thousands. And even if we limit to 70% coverage of a quarter of the proteins, we still have thousands of potential positions. But this is, of course, not relevant for ancient samples. So let's start from the other end and kind of do the opposite. Say, if we have a peptide in a protein, it means that the protein is there. If the protein is there, let's say, very theoretically, of course it's not realistic, let's say we can see the entire protein. So the 19 proteins that were detected in the shy individuals, if we saw the entire full coverage for those 19 proteins, we would have 370 positions. This is enough to make very good clustering of hominins, separating very easily separating very easily modern humans from our close relatives and being able to differentiate Homo sapiens from Neanderthals. The truth lies somewhere in between these two. We don't know exactly where, but let's say 50% coverage of more proteins, 44 proteins that were detected to some extent in these samples. That's not unlikely, and this should be enough to have quite strong power. How do we get there? And I can see I'm running over time, so I'm going to rush through this. And also, we've kind of discussed all of this today already. So let's really quickly go over the proteomic workflow. We start with hypothesis generation and getting samples. We prep the samples. So this is the chipping, powdering, protein extraction, digestion, filtration, and so on, preparation for measurement. Then we acquire data, usually using mass spectrometry, but there are other protein sequencing techniques, and things as simple as like 2D protein gels, Western blots, and so on. That data needs to be interpreted, and we heard a few talks today about the data analysis and database building side of things. I think that is a very important side. Curate the data of a database of proteins or peptides. If it's proteins, then do digestion to create peptides and search for those predicted masses in the data. And finally, we use that to inform phylogenetic analysis and eventually generates archaeological insight. And as a bit of an outsider to the field, I kind of want to give my personal opinion and my perspective on where we are doing very well and where there's a lot of room for improvement, a lot of the things that we talked about today. So I think the most important thing is to establish which questions are answerable and not try to answer questions that paleoproteomics will not be able to answer even theoretically. I think the sample prep is in an excellent state thanks to this community work that we've heard about today and in previous talks. I've seen a lot of good mass spec data. There is room for improvement on low abundance species, so detecting things, non-collagenous proteins, for example, that are in low abundance but are there. And there's a huge room, and we've heard an entire session about it in the data analysis side. The tools, the classical tools like MaxQuant, Mascot, are just not fit for purpose. They are made for detection and quantification, not for sequencing unknown sequences. And we need to take lessons from ancient DNA and authenticate antiquity. Matthew Collins was referring to this earlier. This is really important because this one was one of the big pitfalls of ancient DNA. And because of the way we do this, the more we know what to look for, the more we will find. So working on this side of things is very important. And on the phylogenetics, I kind of alluded to this. We need to take another lesson from ancient DNA and take into account the evolutionary uncertainty. One position, two positions are not enough to determine ancestry. Finally, additional advancements are kind of around the field, non-destructive detection of proteins, Fio's work and others, to detect which samples have proteins and where. Work on biological sex determination that we just heard about, really huge contributions that paleoproteomics can already give insight into. And in my opinion, best of all, this community focusing on early career researchers, open and collaborative, and sharing and optimizing protocols that will lead us there much faster than any of us can do on our own if we focus on the shared success of the field. And also Matthew Collins said it earlier today, if we focus on the shared success of the field, we'll be more successful than if we all focus on ourselves. We can pool data to make the most of precious samples. I've run over time, so I'll just open this and thank you very much.